About complex lipid. So let's start with the introduction to one type of complex lipid that are called shababtid at the end. Remove your phone. Listen. Please, you have to listen. This is you know why you have to listen? Because during this lecture I will remove some slides. I will tell you don't study this slide, don't study that slide. So please. Okay. So if you have to listen and you have to know what you have to focus on, it's a complicated part. I discussed with Dr. Hannah that we need to remove some slides so to make life easy for you. So you have to listen to know which slides are important and which slides should be removed. All right? Now, what are phospholipids? From the term, you can tell that these are lipids containing phosphate. So we have two types of phospholipid. We have the type of phospholipid that contain glycerol, or we have the type of phospholipid that contains sphingosine. So two types of phospholipid. The one that they contain glycerol and the one that they contain sphingosine. But they are polar, okay? And they have fatty acids that are esterified to the glycerol. We'll talk later about the structure. So they are amphipathic. When I say amphipathic, it means that they have two sides. One polar side and one non-polar side. The polar side is due to the presence of the alcohol and the phosphate, and the non-polar side is due to the presence of the fatty acid. So they contain at the same time fatty acids, which are called the hydrocarbon tail, which are hydrophobic, the hydrophobic tails. They contain fatty acid and they are not polar, and they have this polar part which contain the phosphate and the alcohol. So that's why they are considered as amphipathic. So containing two poles, polar and non-polar. And this is important in the structure of membrane. So you know that phospholipid, the most important function of phospholipid, they are structural component of cell membrane. So now this head, we said that we have two fatty acids here. We have glycerol. We have a phosphate group, okay, and we have another alcohol. Okay, so we have the glycerol, which is an alcohol, and we have another alcohol. This alcohol can be serine, can be ethanolamine, can be choline, can be different types of alcohol. All right? Now, we'll talk more about it in a minute. So, the phospholipids are important component of cell membrane. They also play another function, which is the intracellular messenger. Okay? And also they can be attached to other protein in the cell membrane. So the non-membrane bound phospholipids are important component of lung surfactant. You know that in the lung we have what we call lung surfactant, and we have also, they are component of the bile, which is the substance that is released from the gallbladder in order to give the help in the emulsification of lipid in the intestine. Right? So the bile is secreted. It's stored in the, synthesized in the liver and secreted in the gallbladder, and it is secreted to the intestine, and it helps in the emulsification of lipid. They contain phospholipid. So, what are the functions of phospholipid? One, major function, component of cell membrane. Two, they act as intracellular messenger. Three, they are component of lung surfactant, and four, they are component of the bile. All these are functions of phospholipids. Okay? So, we said that there are two classes of phospholipids, those that have glycerol, we call them glycerophospholipid, and those that contain sphingosine, we call them sphingophospholipid. So if they contain glycerol, they are glycerophospholipid, and if they contain sphingosine, they are called sphingophospholipid. But these two classes are structural components of the cell membrane, and they also are involved as signaling molecules. This is sphingosine, and this is glycerol. No structures are required in this lecture. Okay, so you don't need to memorize the structure, but you need to know the structure, understand the structure, the property of the structure. So let's start with the glycerophospholipid. We said that phospholipids are divided into glycerophospholipid and sphingophospholipid. Now we start with the glycerophospholipid. So they contain glycerol, that's why they are called glycerophospholipid. We can call them again phosphoglycerides. Okay, so they are glycerophospholipid or phosphoglyceride, the same. Now sometimes, because we're talking, we use phospholipid, but phospholipid, the word phospholipid is not enough to indicate if I'm talking about a glycerophospholipid or sphingophospholipid. Okay, so that's why it's better to say glycerophospholipid or sphingophospholipid. So the glycerophospholipid constitute the major class of phospholipid. They are all derived from a molecule which is an acid called phosphatidic acid. What is phosphatidic acid? Phosphatidic acid is made up of the diacyl glycerol. What do we mean by diacyl glycerol? It is the glycerol containing two fatty acids. It's a glycerol where you have attached to carbon number 1 and carbon number 2 fatty acids. And also it contains a phosphate group at carbon number 3. So at 1 we have fatty acid of glycerol, at 2 we have another fatty acid, and at 3 we have a phosphate. This is called phosphatidic acid. It is the simplest type of phosphoglyceride, and it is the precursor of all the other types of phosphoglyceride. So the backbone structure of a glycerophospholipid is phosphatidic acid. So the phosphate group of phosphatidic acid can be esterified to another compound containing an alcohol group. So to make the glycerophospholipid, the other type of glycerophospholipid, we need to add another alcohol on phosphate. This part, by its own, it's called phosphatidic acid. This is glycerol, this is the fatty acid, this is the phosphate, okay, which is in yellow, okay, the red is the glycerol, and this part is an additional alcohol which is attached to the phosphate, and this type of alcohol make the different types of glycerophospholipid. So, if you have phosphatidic acid plus serine, the phosphoglyceride is called phosphatidylserine. If you have phosphatidic acid plus ethanolamine, and if this head is ethanolamine, we call it phosphatidyl-ethanolamine, and it's also called cephalin. If this head, like here, it is choline, so this structure is called phosphatidylcholine, and it is also called lecithin. And you have to know lecithin very well because it is involved in the maturation of the lung of the fetus. If the baby, you know that the baby may have sudden death after birth, and this is due to immaturity in the lung. So lecithin is one of the components that should be present in the lungs to have mature lung. And if this head is inositol, we call it phosphatidylinositol. If this head is glycerol, we call it phosphatidylglycerol. So all these types of glycerophospholipids depend on this head. What head we have, but they all contain phosphatidic acid. Clear?
Yeah.
So now here just to recall back what's the difference between ester and ether group. You know very well that ester means that you have a carbonyl group that is involved in the esterification, and here you don't have a carbonyl group, so it's an ether linkage, which is an OH here. Okay, so you should know the difference between the two. Now, what type of linkage we have here with the fatty acid to glycerol? Ester. So it's an esterification of two fatty acids, one on carbon number 1 and one on carbon number 2. At carbon number 3, what do we have? Phosphate. This part is called phosphatidic acid, and this is the blue one here is the head. This head that we were talking here about. If this head here it is serine, we call it. If this head is ethanolamine, if this head is choline, which is lecithin, and this is phosphatidic acid without a head. You see, they have only phosphate group here. So only phosphate, glycerol, two fatty acid, phosphate is phosphatidic acid, and then the phosphatidic acid is the precursor of all these types of phosphoglyceride. You have phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylglycerol, phosphatidylglycerol, and many other types of glycerophospholipid, but these are the most important types. What do we need to know about these types? You just need to understand the structure of a glycerophospholipid, what's the precursor, you have to know what's phosphatidic acid, and the different types of glycerophospholipid. Without writing the structure, you should know that, for example, phosphatidylcholine is composed of phosphatidic, like this one. Okay? No need to know the structure. No need to know these details. All right? Although you know what's a serine, right? What's serine? It's an amino acid. But why I consider it here as an alcohol? It contains OH, CH2OH. The R group of serine is CH2OH, so that's why it can be attached to phosphate. Another type of glycerophospholipid that is much more complicated. Why it's much more complicated? It's called cardiolipin because it was first isolated from the heart, but it's present in many other cells. It's found in the bacteria, and it's present in many cells in the inner mitochondrial membrane, and it is part of the play role in the respiratory complex of the electron transport chain. So it's involved in the electron transport chain. But what about this cardiolipin? It is composed of two glycerophospholipids that are attached by a glycerol. So look what you have here. You have one and another one, and they are both attached by a glycerol. So it's a complicated structure, no need to know about it. But what you have to know about that the cardiolipin is antigenic, and it is recognized by the antibody that are raised against Treponema pallidum, which is the bacteria that causes syphilis, which is a sexually transmitted disease. So this bacteria can cause the antibodies of this are, can recognize cardiolipin as antigenic. Now we have a special type of glycerophospholipid are called plasmalogen. Why they are special type? What did we say about the carbon number 1 here? What do we have here? We said we have fatty acid. Do we have fatty acid here? No. So there is an exception. There is no an ether. There is no esterification ester. There is an ether linkage here. Why? Because instead of having a fatty acid at carbon number 2, we have an unsaturated alkyl group. This is an unsaturated hydrocarbon group. Unsaturated because we have double bond. So this is not a fatty acid here. It is an unsaturated alkyl group which is attached by an ether linkage to the core of glycerol. Okay? Now, okay, so what do we call it in this case if we don't have a fatty acid? What will happen? Let's say here we have ethanolamine, right? Normally it is called phosphatidyl ethanolamine. Now in this case, what do we call it? Normally it is phosphatidyl ethanolamine. To differentiate it between the phosphatidyl ethanolamine, so the one that contains fatty acid, and the one that does not contain fatty acid, which is very abundant in the nerve tissue, we put an A instead of Y. So phosphatidyl ethanolamine, the DA, here DY. So DY means that you have fatty acid, and when you see DA, you know that it is not a fatty acid on carbon number 1, it is an unsaturated alkyl group. Now sometimes, just to finish this idea, sometimes in some reference book they call it phosphatidyl ethanolamine plasmalogen. They add plasmalogen instead of changing this letter. Yes?
Yes. Does the plasmalogen such the non-polar property that the regular phospho?
Yeah, they do. This is a good question. Thank you for asking this question. Yes, still we have non-polar ends. These are non-polar. So the non-polarity is coming from the hydrocarbon chain. And in both cases you have hydrocarbon chain. These are non-polar. Okay, so still it is yes, we do have the non-polar side, which is the fatty acid and the alkyl group here. Okay, so we have also another example is phosphatidylcholine, which is very abundant in heart muscle, and also we have other types of these plasmalogen that are present in mammals. A third example of a plasmalogen, it's not a plasmalogen actually, because in some reference book they call it plasmalogen, but it's different types of glycerophospholipid again here. So instead we have here a saturated hydrocarbon chain. Okay, it is saturated this time. And another exception is that instead of having a fatty acid here, we have acetyl group. Okay, we have an acetyl group. And here the polarity, the non-polar side is less, because we have only one non-polar side instead of two. Okay, instead of having two groups, you have one group here. So what about this platelet activating factor? So you have hydrocarbon chain here, you have acetyl group on carbon number 2, and the importance of this structure called PAF, or platelet activating factor, that it binds to surface receptor and it triggers potent thrombotic and acute inflammatory events. Okay, platelet activating factor can activate inflammatory cells and it mediates hypersensitivity and anaphylactic reaction. It causes platelet to aggregate and neutrophil and alveolar macrophage to generate superoxide radicals. So this compound is considered as the most potent bioactive molecule It can cause effect at a very low concentration, which are 10 to 12 mole per liter. Yes?
You said you have an acyl group instead of fatty acid, isn't instead of glycerol?
No, this is glycerol. This one is glycerol. And normally on glycerol and glycerophospholipid, we have one fatty acid here and one fatty acid attached to carbon number 2. And this is carbon number 3, where you have a phosphate and an alcohol. Now the exception here is that you don't have fatty acid here. You have a hydrocarbon chain, saturated one, and here you have acetyl group. So the difference is in the structure of the carbon number 1 group and carbon number 2 group of the glycerol. But here it's the same. Here, no change. So that's why in some reference they consider it as, it is a phosphoglyceride. Why phosphoglyceride? Because it contains phosphate and it contains glycerol. So they are all called phosphoglyceride, but exception in some structures. Now we move to a new class of lipids that they don't here contain glycerol. They contain another alcohol called sphingosine. So that's why they are called sphingolipids. They are also important component of the cell membrane. Although they don't contain glycerol, but they have similar structure to glycerophospholipid, and also they have a hydrophilic region and 2 fatty acids attached to the sphingosine this time, and they differ primarily in the nature of the hydrophilic region. So the difference is not in the hydrophobic region, the difference is in the hydrophilic region, the difference compared to phosphoglyceride. Okay, so they are also component of the cell membrane. What's the difference here? In sphingophospholipid, we have sphingosine. Sphingosine is a C18 alcohol. C18, it means that it's composed of 18 carbon. It's an alcohol. But at carbon number 1, we have here esterified a phosphocholine. So this will form what we call sphingomyelin. I'm sure that you heard about sphingomyelin. It's a component of the myelin sheath. In the multiple sclerosis disease, you have degradation of the myelin sheath, as you know. So this is an important component of the myelin sheath. It's present in the myelin of nerve fibers. So you have then sphingosine, the one that is in the green, and additional fatty acids. So you see, that's why I said that the hydrophobic region are similar to the glycerophospholipid, because they have 2 fatty acids again here. This is the hydrophobic region. But the alcohol is not glycerol, it is sphingosine. When you have an extra fatty acid here, we call this structure, the one labeled in red, ceramide. So what is ceramide? Ceramide is a sphingosine plus fatty acid. So this is carbon number 1. We said that when you have a phosphocholine, why phosphocholine? Because this is choline in blue, and in white it's phosphate. So this is phosphocholine. The whole structure is called sphingomyelin, and it is the component of the myelin sheath. It's present in the myelin of nerve cells. It protects the neuronal fibers of the central nervous system, and you know the importance of this structure in the nerve fibers. Okay, is it clear? So what is sphingomyelin as a summary, and then you ask your question. Sphingomyelin is a sphingolipid because it contains sphingosine. Sphingosine is an alcohol. Sphingosine plus fatty acid form ceramide. Sphingomyelin is ceramide plus choline. And this choline is attached to carbon number 1 of sphingosine. Yes? No, I answered the question? Okay. So this slide is not for study. I don't want you to study the synthesis. So you can delete this slide, but some people would like to know how they are synthesized. So if you want to know how they are synthesized, this is the answer. You don't know. You don't want to know. I know. We have 2 ways of synthesizing the phosphoglyceride. You know that in the glycogen synthesis we used UDP. What was UDP? Okay, what is CDP? Okay, great. This is a carrier. So you can add the component of phosphoglyceride are diacylglycerol and an alcohol plus a phosphate, right? So you carry the diacylglycerol on a CDP and then you add the alcohol. This is one way. Or you can carry the alcohol on ACDP and then you add diacylglycerol and you form the phosphoglyceride. So this is, don't worry about this slide. And also, this is what I just said, the phospholipids are synthesized in the endoplasmic reticulum and they are transported to Golgi apparatus, then to the membrane of the organelle or the plasma membrane, and they are secreted by exocytosis. So don't worry about the synthesis of phosphoglyceride. But you have to worry about the slides. And you have to pay attention because this is part of your, it's very important for you. So we mentioned at the beginning that some fetus, newborn, they may have sudden death, and mainly it may happen to a preterm baby, who did not complete the 9-month period. So this is caused by a problem in the maturity of the lung. And this maturity of the lung can be determined by two components. These components are the one that we explained, lecithin, which is a glycerophospholipid, and sphingomyelin, which is a sphingolipid. The two structures that we explained at the beginning of the lecture. And to know if the lung is mature, you have to ask for a test called LS test in the amniotic fluid. What is LS test? LS test means the ratio of lecithin over sphingomyelin. When it is more equal or more than 2, there is a maturity. When it's below, it means that there is no maturity. And this synthesis it should occur in the pneumocyte. Why it's an indication? Because when the gestation is 32 weeks, the pneumocyte should have the certain amount of these phospholipids in the pneumocytes. Okay, what will happen if you have a problem with the level, with the ratio? What do you? How can you sort out this problem? So the study showed that they give the mother glucocorticoid shortly before delivery, and this will stimulate the production of these phospholipids, and they may have they may stop the problem in the lung. They may help in the maturity of the lung, and also they may also give the synthetic surfactant because to prevent a disease known in the infant known respiratory distress syndrome, RDS. This disease is not only in the newborn, it may occur also in adults, and this can be due to a damage in the pneumocytes, due to a destruction that can be caused by an infection or trauma. So it may occur in adults, not only in infants, due to problem in the lung. So the lecithin is the major also component, as we mentioned at the beginning of the lecture, of these lung surfactant. So lecithin is an important component of lung surfactant. What is the surfactant? The surfactant is a mixture of protein and lipid, and it's responsible for lowering the surface tension at the alveolar air fluid interface. So this component is important, okay, in the lung. That's why we mentioned one of the function of glycerophospholipid is it is a component of the lung surfactant. So we explain here the structure. We mentioned the structure before. It contains palmitic acid. The fatty acid are palmitic acid, esterified at position number 1 and number 2, and the phosphocholine esterified at position number 3. We mentioned the structure before. Question on the issue that the lungs are mature. You have to know what is the RDS, respiratory distress syndrome, and how we sort out this by either giving the synthetic surfactant or by giving glucocorticoid. And we talk about lecithin as a major phospholipid in the surfactant, and what's the importance of the lung surfactant and the structure of this. Yes?
So if the number is less than 2,
Yeah.
that means it's not mature yet.
Yes.
So the problem will be from what exactly?
It will be from the embryogenesis. We don't know what's the problem, but the problem, the essential problem is the level of these lipids in the lung. So that's why we have to measure them. Okay? Now, also, we talk about the degradation of phospholipid. The degradation, I want you just to know from this slide that the degradation of phospholipid occurs in the presence of enzyme called phospholipase. Now, these phospholipases can be secreted by toxin and genomes, okay? And they dissolve. What happens when they secrete phospholipase? The phospholipase will degrade the phospholipid. So they will degrade, they will dissolve the cell membrane, and this will allow them to spread the infection. So they give these substances, these enzymes, to degrade the phospholipid and to dissolve the cell membrane in order to allow the spreading of the infection. It's stop and take the attendance. Okay? To my names. Okay? Now, let's see how these phospholipases act. Remember the structure of glycerophospholipid? We have glycerol, one fatty acid attached to carbon number 1, another fatty acid attached to carbon number 2, a phosphate group attached to carbon number 3, and then an alcohol attached to the phosphate group. This is a glycerophospholipid. We have different types of phospholipases. Phospholipase A1 cut here. So it removes the fatty acid. Phospholipase A2 cut here. Have you studied the eicosanoid structure synthesis, arachidonic?
Yes.
Dr. Rasool explained it to you, right? He said that arachidonic acid are released from phospholipid, right, by the effect of phospholipase A2. And he said that arachidonic acid are transformed into prostaglandin, leukotriene, and thromboxane. So the arachidonic acid is released by this type of phospholipase. And he said for sure that this enzyme can be inhibited by glucocorticoids such as cortisone. Right? So if you want to inhibit the synthesis of prostaglandin, you inhibit the release of arachidonic acid from this space, and you inhibit by inhibiting the enzyme phospholipase A2 by glucocorticoids. True that he explained this in the lecture. So, okay. Now these two enzymes will release the fatty acid. The enzyme phospholipase C will remove this part. What do we call this part?
The alcohol.
Yeah, no, it's not only the alcohol. The phosphoalcohol. So the phospholipase C will remove the phosphoalcohol. So if I ask you adding phospholipase C to this glycerophospholipid will release which product? Diacylglycerol and phosphoalcohol. So if I have choline here and I add phospholipase C, the product will be diacylglycerol and phosphocholine. If I add phospholipase D on phosphatidylcholine, phosphatidic acid, because this one is called phosphatidic acid and choline. So according to the type of phospholipase, you have the types of products that are released from the hydrolysis. Right here. Now we move to glycolipids. What are glycolipids? So from the term you can tell these are lipid containing carbohydrate. Okay, they are like sphingomyelin. They are derived also from ceramides. So glycolipid contain ceramide. Okay, they are essential component of cell membrane, and they are present in large amount in the nervous tissue. They are located in the outer leaflet of the plasma membrane, and they interact with the extracellular environment. So that's why they are important. They play a role in cellular interaction, such as cell adhesion, cell recognition. They play a role in the growth and development. Okay, this is the function of these are the function of glycolipids. So what do we have in the glycolipid? Or glycosphingolipid. As I said, the sphingosine with the fatty acid will form ceramide. So a glycolipid contains a ceramide plus sugar. A glycolipid is a ceramide plus sugar. They do not contain phosphate. So that's why they are not phospholipids. So I may ask in the exam, which of the following lipid does not contain phosphate? And if I put glycolipid, you have to know that this is glycolipid. Okay? They do not contain phosphate. So sphingomyelin contains phosphate, phosphoglyceride contains phosphate, but glycolipid, they do not contain phosphate. You have only ceramide and sugar. And this sugar is attached to the ceramide by an O-glycosidic bond. So let's see. We have two major types of glycosphingolipid. We have the neutral glycosphingolipid and we have the acidic glycosphingolipid. In the neutral glycosphingolipid, they are the simplest type, and they are not charged. They are composed of ceramide and a monosaccharide, and they are called cerebroside. So if you have a galactose here and a ceramide here, this structure is called galactocerebroside. If you have a glucose here and a ceramide here, this structure is called glucocerebroside. So according to the monosaccharide that is attached to the ceramide, you give this the name of this structure, either galactocerebroside or glucocerebroside. So they, as I said, they are present in the brain and the peripheral nervous tissue, and they are present in high concentration in the myelin. As I said, we have two types: the neutral, which is not charged, and the acidic. In the acidic, we have two types or two subtypes. We have the gangliosides and we have the sulfatides. So neutral are only monosaccharides containing monosaccharides, so they are called cerebroside. Acidic, we have two subtypes, ganglioside and sulfatide. But they are both acidic because they are both charged. Okay? We have the charge here. In the first one, which is the ganglioside, we have a ceramide, but we have an oligosaccharide. You see here? This is an oligosaccharide attached to the ceramide by an O-glycosidic bond. But in the oligosaccharide, we have an important component which gives the negative charge to the structure. It is an acetyluraminic acid, or called sialic acid, because of the presence of the carboxylate group here. Okay, so what is ganglioside? Ganglioside is composed of oligosaccharide plus ceramide, and in the oligosaccharide we have an acetyluraminic acid. Whereas in sulfatide, again we have ceramide and a monosaccharide like the cerebroside, but in the monosaccharide we have a sulfate group. So one of the OH of the monosaccharide is sulfated. That's why this structure in this case is called sulfatide, and sulfatide are present in the brain and the kidney. So in glycosphingolipid, we have the neutral glycosphingolipid, which is the galactocerebroside or the glucocerebroside. So they are called cerebroside, composed of one monosaccharide and ceramide attached by an O-glycosidic bond. And the acidic types, we have two subtypes, the ganglioside and sulfatide. The ganglioside is composed of an oligosaccharide attached to ceramide, where an acetyluraminic acid is a component of the oligosaccharide. And in the sulfatide, you have a ceramide and one sugar, and this sugar is sulfated. That's why it's called sulfatides, and it's important in the brain and the kidney. These are the complex lipids. They are complicated, but please try not to complicate your life and make them easy. So forget about the synthesis. I just want you to know the component of this structure and their function. Okay? And also, so forget about this one. This is the synthesis. All right? Forget about slide 23. Yes?
Can you sulfify?
Yeah.
The monosaccharide should be galactose.
No. What the major one is that's why we give it as an example. So you should know the classes of sphingolipid, that the sphingomyelin contain phosphocholine, and in the cerebroside we have galactose or glucose. In ganglioside we have an oligosaccharide containing an acetyluraminic acid. This is what I want. Now we have a group of disorders called sphingolipidosis or lysosomal lipid storage disease. Okay, what are these diseases? You remember the glycogen storage disease?
Yes.
So it's a group of disorder related to the metabolism, synthesis or degradation of glycogen. Right? Here, same, you have problem in the synthesis or degradation of these sphingolipids. They are collectively called sphingolipidosis. So we know that the sphingolipids normally are released when the membrane is degraded, and they should be digested in the endosome after fusion in the lysosome. They are digested by lysosomal enzymes. So these enzymes in the lysosome will degrade the sphingolipid. Now we may have deficiency in these enzymes that degrade the sphingolipid, and that's why we will have a disease called sphingolipidosis. And it shares some characteristic with the disease called eye cell disease, which is this disease is caused by a deficiency of the enzyme that transport. Yes. So you know about this enzyme. So what I want you to know about the disease is three only. Okay, just three examples. One is Tay-Sachs disease. It's characterized by deficiency in the enzyme hexosaminidase A. It's the enzyme that releases the hexose. After degradation, you know, we have sugar attached, right? So this will cause accumulation of ganglioside, and the symptoms are cherry red spot in the macula. It may cause blindness. It may cause also early death before the age of 2 years. Another disease is called Gaucher disease. It is caused by accumulation of glucocerebroside due to deficiency in the glucocerebrosidase enzyme. So here you have problem in the organ. There is hepatomegaly, splenomegaly, and problem in the count of the cells. The third one is Niemann-Pick syndrome or disease. It's characterized by accumulation of sphingomyelin because of deficiency of sphingomyelinase. You may have a cherry red spot in the macula, but here there is more problem in the organs and early death may occur. So if you look at the aspirated bone marrow of a patient with Gaucher disease, you can see the accumulation of glucocerebroside here. You have elongated lysosomes. They are filled of abnormal amount of glucocerebroside because of the deficiency in glucocerebrosidase enzyme. If you look at at this slide here, it will show the accumulation of sphingomyelin because of a disease called Niemann-Pick disease. So there's deficiency in sphingomyelinase enzyme. So also you have, as I said, enlargement of the organs because they accumulate in these organs, and they cause mental retardation because you know that these are components of the brain and early death may occur. So you have question to test yourself. RDS, do it at home. And it is another example, a third example. No need for the answer. You have to know the answer. If you don't find the answer, send me an email.